Hi,
Hi. How are you?
fine, thank
Fine,
you.
thank
And
you.
you?
I'm fine too. Do you listen well to me?
Sorry, I
Sorry, I
don't
don't
understand.
understand. If can you hear me all right or so? Can you hear me right or so?
It's
It's
working,
working,
but
but
a little
a
bit
bit
laggy.
laggy. Hi there. How are you? I cannot listen to you. Ah, no. Can you hear me properly?
Uh, Not
Not
really,
really,
but
not
I think
really. Okay, let me check. One sec.
I think I
I
will
will
shut
shut
down
down
my
my
camera
camera
to have
to
a
have a
better
better
connection.
connection.
How's the
How's the
audio?
audio? Maybe it's me, but I don't know.
I will
I will
try.
try. I think my connection is fine, but can you hear me properly now?
Yes, a
Yes,
bit
a bit
better.
better. Yes. Okay.
I
I
think
think
it's
it's
my
my
connection,
connection.
it's not really.
Okay. We are missing still one person, right?
Yes.
Yes.
Oh,
Oh,
is
it's
here.
here. Oh, hi. So, is the three of you the attendees to the class, right? Hi. Hi there.
Hi.
Hi. Okay, first of all, let me introduce myself because it's the first time we meet each other and we will be meeting the 4 next sessions, including today. So, my name is Lucia and I am going to be explaining the topics about disease networks, also the 2 next sessions that would be around biological data and bioinformatics, and then the last one that would be vocabularies and mappings that will be related with disease terminology and classifications and ontologies and all that kind of data that codify diseases. Okay, so if you can just tell me your names and your backgrounds, so I can more or less know what I have to tell you about the more biological stuff and what you already know. I would very much appreciate.
Okay, I
Okay, I
can
can
begin if
begin
you
if you
want.
want.
Uh, I
I
have
have
a
a
professional
professional
background
background
in
in
medical
medical
imaging,
imaging,
MRI,
MRI,
scan,
scan,
in
in
biological
biological
field
field
that
that
I
I
have
have
some
some
basics,
basics,
but
but
I'm
I'm
not
not
specialized in
specialized in
it.
it.
And I
And I
have
have
some
some
basic
basic
knowledge
knowledge
on
on
data
data
analysis,
analysis. Mm-hmm.
and it's it's
It's
all for
all
me.
for me. Okay, and can you repeat me your name because I didn't understand?
Oh
Oh
yes,
yes, yeah,
I'm
I'm
Robin.
Robin. Robin.
Yes.
Yes. Okay. So the next one.
Okay.
Okay.
My
My
name is
name is
Carlos.
Carlos. Hi, Carlos.
I'm
I'm
studying
studying
the
the
medical health and medical
medical
data
data
analytics
analytics
from
from
UIT,
UIT,
and
and
I was
we're
studying
studying
the
the
first
first
year
year
at
at
Germany
Germany
in
and
Erlangen,
Erlangen,
in the
in the
University of
University of
Erlangen.
Erlangen.
And
And
my
my
background is
background is
in
in
biomedical
biomedical
engineering
engineering
too, and
too,
I
and I
have
have
a
a
professional
professional
background
background
also
also
related to
related to
3D
3D
modeling
modeling
for
for
medical
medical
images.
images.
So I
So I
have
have
some
some
knowledge in
knowledge in
biology,
biology,
but
but
it's
it's
not my
not my
strong
strong
point,
point,
I
I
have
have
to
to
admit.
admit. No problem, no problem. So,
hi,
hi.
I'm I'm Abhinaya,
Abhinaya,
like
like
can
can
call me
call me
Abhi.
Abhi.
So
So
basically
basically
I'm
I'm
from
from
India
India
and
and
I'm
I'm
doing my
doing my
masters
masters
in
in
Germany.
Germany.
my specialization is like medical engineering, health and medical data analytics. So same as Carlos, like I have a good background with biological knowledge, and technically we are like towards engineering in the field of medical. So
Okay,
that's
makes
it.
sense. For myself, I studied biotechnology and I specialized in computer biology, computational biology. So if you think something it's not clear because I have given that for understood, just tell me and we will recapitulate and see what's everything, okay? Because sometimes I just go and say, I don't know, DNA or RNA, something that maybe for me is super clear, but not as much for you, okay? So just stop me whenever you want, and we will see. I think it's not going to be an hour or 2 hours, but like an hour and something. Okay, so that's good because I think diseases networks are not so big deal. Having you learned the two previous sessions, no, three previous sessions that Masi did the other days. So I think if you understand those concepts, disease networks are just a special case for that. Okay, so now I'm going to share my screen with the presentation. Let me 1 second. Now you're seeing my screen, right?
Yes, it's working.
Perfect. So let's start. This session is called Disease Networks, and today will be structured in 7 points. We will first introduce the topic. We will then talk about the human disease network, that is the most important approach and the one that lead to the others around diseases networks. Then we will explain a special point about human disease network, that is the human symptoms disease networks. After those points, we will go with other disease networks based in other kind of information. We will see some approaches, advantages and utilities of these approaches, and then I will explain you one use case that is TISNet, that I think that Alejandro in the first sessions already mentioned it, that is our particular project around diseases networks. And finally, I will just point out some of the other applications and projects that are being around all of these concepts. And another thing that I haven't mentioned is that my English is so-so, so if you think something is not clear, just tell me and I will try to re-explain it with some other words, okay? So, let's start with the introduction. I would like to ask you, what do you understand for a disease? What do you think, in more philosophical ways of thinking, a disease is? Anyone?
It's like a physiopathological problem.
You would define a disease like a physiopathological problem, and that's all. Like, I mean, would you puntualize something else, or I don't know? but it's quite accurate. The term physiopathological involves the word physiology, that is the correct functions of the organisms, and pathological is the not working correctly those processes. So it's quite accurate. But do you want to say something more, or?
It's just like normal function of the body is not working correctly, so there is some problem inside that.
Yeah, that's yeah something.
Something which is not happening in a usual way.
Yeah, yeah, but basically, so that would be like our way of thinking. To say is something that is not working correctly, as Abby said. Um, we can specify a little bit more that definition with saying that it can be caused by multiple things, and it can be derived from multiple traits, and also those not going okay states or physiopathological processes can derive not only in one disease or a disease, but also in multiple phenotypes. Okay, so I have mentioned some concepts here that are diseases, phenotypes, physiological processes. What I want to say is that when we talk about diseases, I'm sure that you all know, because you are all specialized in the medical field. We are not talking about a very concrete thing, a very particular thing, but something quite abstract and that requires of multi knowledge domains. Okay, it's not just one domain. Here in this first slide that you can see, we have several layers, let's say, of things that are related with diseases. If we go down in the boxes, we can find the genetic networks, okay, with the genotypes and transcription factors. If we go a little bit up, there are signaling networks that can be on signaling proteins or mediators as other biomolecules, as can be, for example, microRNAs or those things that are telling other biomolecules to do or to be involved in other processes. Then if we go one step up, we can find metabolic networks that those are when we talk about metabolites and enzymes that are codifying all the metabolisms of an organism. And then upper, we can find the phenotype and the external factors. Sorry. So the conjunction of all of these networks, all of these information or let's say processes, is what we can output as a disease. Okay, maybe some things are dysregulated here, and some things are being inputted here, and finally what we can find is that in the global output there is something that is not working properly, as we have commented, deriving to what we can call a disease. So this is just a schematic diagram showing how is the typical coordination between those genetic signaling and metabolic networks, and how all of these networks generate a phenotypic response. the combination of the different phenotypes or the aggregation of the different signs and symptoms is what we can, let's say, understand as a disease. All of these networks, also genetic, signaling, and metabolics, we can find them in the cells of the organism, in all cells. Maybe in some cells of the organisms are okay, they are not dysregulated, they are fine, but maybe they are focused in one tissue or in one type of cell, and we are already developing the phenotype for those cells, and that is causing the disease, okay? If we have a problem in any of the levels, that would be a modification. It can be okay. We can be okay still and not showing to the outside a phenotype, but if the concrete the particular point that is being modified is a key point for some process, it will be a matter of a disease, okay? So yes, as I was saying, any defect at any of the levels of the processes can lead to a disruptions of the adequate response of the organisms, what will lead us to disease states. This vision is what we can say that is studying the disease as a systemic defect, and it has much to do with what we want to study today, okay? This would be an example, a particular point in the previous schema. That would be the map of the complex nature of insulin resistance and defective metabolic homeostasis. Okay, if you go deeper in the figure, you can see that there are some arrows in red and some arrows in green that represent whether effect is being positive or negative corresponding to the different inputs that we have, okay? All of the combination of the inputs and the regulation of the different biological processes would output that phenotypic state that can be or not a disease. If it is a normal phenotype, it won't be a disease, but if something is going wrong, it will, okay? So all of these information and knowledge that we have about diseases has been revolving around a concept that is network medicine, that has become especially relevant in the previous decades due to the large amounts that we have now of biological data. I'm sure that you all know that some decades ago there wasn't as much information, the science wasn't as evolved as it's now to acquire all of this knowledge from processes and from, for example, sequence in DNA or in RNA or proteins or whatever. And now we can access that knowledge just by typing it in the navigator. So it has become a really relevant field because of its impact in medicine, the knowledge that we can obtain by relating all of the information that is available now. Okay. this field of medicine, that is what we will be talking about today, because disease networks are involved in network medicine, refers to the capacity of being able to put all together, okay? That is what we call holism, that wants to study all of the diseases at once, not by separate, okay? So it has the capacity not only to approach one human disease and study its molecular complexity, but study also the molecular relationships between what we call apparently distinct pathophenotypes. Pathophenotypes, it's a phenotype that is showing that something is not okay, what we call a disease, okay? As I said, this approach of network medicine is not working with molecular reductionisms. That was what people and scientists tried before. Like, I mean, before all of this information was available, researchers usually tend to focus in one disease and study its complexity, but didn't go and see what similarities were with other diseases and study both at the same time. Or maybe yes, but in those cases that diseases were quite similar or were in the same class of diseases, okay? Not to see if one disease of the I don't know, metabolic system was related to one disease like lung cancer, for example, okay? No, they didn't try to analyze the relationships between diseases, but study just how the molecular processes beside a disease were occurring. So is now that we have all this data that we can turn things around and try a holistic approach and say, okay, I have this molecular information about the disease. I want to see if it is related to another disease. If it is related, why? Which target is involved in both diseases? So I'm talking about targets. Which drug I can use to prevent or to treat that disease, okay? Yeah, and so as we will see later, there is an actual fact to see that the relationship between diseases and to use the same drug between diseases when they are quite similar or they share some molecular processes or just some processes, okay? Maybe they share phenotypes and that phenotype is being caused by something and the drug is treating such phenotype, and you know that that drug is being used for that symptom in one disease and maybe you can use it for the other. Okay, we will better see later. So yes, this slide you can see here what I was talking about, the molecular reductionism that was approached the last century, let's say, and now that has evolved to genomic holism, that tries to develop around all of these networks in the cells or in the organisms that can be altered and that can be the cause of disease or maybe the combination of the multiple of them, okay? We have mentioned a word that is pathophenotypes, that as I said, are the what we call normally diseases that come from the combination of multiple intermediate phenotypes that are derived from some dysregulations, maybe in the genome, transcriptome, or proteome, in combination with the environmental perturbations that can affect the body. Okay, I'm almost sure that you all know what genome, transcriptome, proteome means, but I'm going to quickly enumerate them so you have it clear in mind. Genome refers to all the sequence of DNA in the organism. You know that from DNA we have RNA and RNA, no microRNA that we will later talk about it, but it's a type of RNA, but not in RNA. Okay, so the transcriptome involves all those mRNAs, and then the proteome involves all of the sequence of proteins in the body of amino acids. Okay. So as I was saying, all of the diseases are usually a combination of all those points, not only the modification of one, as for example Mendel thought. Those Mendelian disorders that we used to think that were caused only by the mutation of one gene. That is quite unusual, let's say. Okay, normally the diseases are caused not only by one modification or the dysregulation, but by several levels of dysregulations. Okay. Yes, talking again about network medicine, these figures summarize it pretty well, because we can see at the left in A theoretical human disease network that illustrates the relationships amongst all of the genetic and environmental elements that determine the phytophenotype that is in red. Okay, as you can see, we have two here. We have the primary disease genome, then here we have the secondary disease genome, and here the environmental determinants and the intermediate phenotypes that we saw in the last slide, that all in combination can cause, not cause, but can be summarized, let's say, in pathophysiological states that lead to a pathophenotype that is a disease. One particular case is the one that we can see here in B, that is one network related to particularly sickle cell disease. Do you know that disease? The one that is the anemia, that where the red cells have different form, because the beta globin gene is mutated, so the red cells have another form, and they cannot carry the oxygen as they usually do. Okay, so here you have in red that gene that I was talking about, that is the beta globin gene. But although this is the thing that we see, that we researchers find first, because it's the most evident disruption, there are other elements or determinants that are causing that pathophenotype. Okay, for example, in gray you can see some disease-modifying genes, depending on how the genes are expressed, the primary gene, let's say, will be more or less. Then we have in yellow the intermediate phenotypes that we were talking about, okay? Some environmental determinants in green, and finally in blue, those pathophenotypes, okay? I don't want to extend much these concepts, but I think it's important to clarify some terminology. And here, what we can see is the inputs, let's say, that we use when we are building networks in network medicine, and the outcomes, the advantages we obtain from constructing those networks, okay? Disease networks, or we would say biological networks, because as we will be explaining today, there are not only relevant disease networks, we can find also other types of networks that can help us to building disease networks, okay? These biological networks that I was saying can be built based on multiple data sources, because as we will be explaining the next sessions, there are plenty of biological databases, and they are still growing. They are getting bigger and bigger, and the information is being more accurate. And also, we have numerous kinds of information, from genes to relationships between genes and diseases, and also sequence and arrays of genes expressions. Okay, we will. I think you already saw some of them in the first presentation class on the second, but we will stop in the most important biological databases on next Tuesday. So as you can see, here we have, for example, hospital networks, that are those corresponding to patient-structured information, depending on the disease that we are studying, or if we are studying all the diseases at the same time, this type of networks would be one way or another. Then we have tissues networks, how the different cells, tissues are being expressed one gene or another, okay, or if some disruption is taking place in one tissue but not in other, okay. The cellular networks involve all of the regulation and communications of the different biological compounds in the cell. Molecular networks, if we are talking more about physical interactions, for example, protein-protein interactions that we will see later. And then in the most down layer, we have genes that also we can build genetic networks where we can relate genes based on different things. For example, the diseases to which they are related, okay? All of these networks can help us to several tasks. For example, identifying biomarkers and knowing where to sequentiate a gene of a person that we think might be porter. I don't know if that's the word of in English, the portador of the disease. Also, we can find for different therapeutic targets in the networks and how to diagnose the diseases. As we were talking about, to diagnose ICs, we will see the pathophenotypes and determine the combination which is the more accurate disease that is associated to that pathophenotype. Okay. Also, diagnosis, as I said, and treatment. As we will see, all the drug repositioning domain, for example, and the drug discovery can be also addressed using this kind of approaches. And also the classification and subtyping of diseases that we are very familiar, let's say, with the type of classifications that put diseases in different groups based on the anatomy that they are affecting. But molecularly talking, that is not the most accurate in some points way of classifying disease. And finally, here you can see also another outcome of this type of networks that are symptoms networks, not only disease networks, but we will later see it more specifically. The things that I have already told you about network medicine, that it can reveal some potential nodes and pathways using a graphic spectra. Okay, this comes from disease network construction that will come from biological data mining, and in first step it derived from the disease and they're studying the data collection that we obtain from that. Okay. We will later talk about this, but what you can see here is that drug discovery and drug development has evolved during the years, and now all of the clinical states, the clinical trial states are more defined than 20 years ago, let's say, and it's all a hard process, and now there are not being as much as drugs developed as it were before, because all of the security procedures are higher. I mean, they are more restrictive, let's say. And what you can see in the last figure, in the one in the top, in the bottom, letter C, is the different kind of biological networks that we were talking about. The metabolites, the genes, proteins, the proteins interactions, also the metabolic interactions, gene regulation, gene interaction, cell signaling. Loads of them, okay? And all of the combination of the different different networks, we will evolve in analysis if some of the process or links, let's say, are dysregulated, okay? This concept of network medicine is based on graph theory that I guess that you now know a lot about after Masi explained to you the last sessions. Graph theory is the field of mathematics and computation that has achieved a lot of breakthroughs to biological networks. All of biological, or most of biological networks have the nature of a complex network. A network, as you know, from the mathematical point of view, is defined by vertices or nodes and edges or links. Vertices are linked by edges if they share or something, or if they have a relationship. In biology, these nodes may represent genes, proteins, metabolites, RNA molecules, also diseases, that is what we are going to see today, phenotypes, and the edges are the connections or interactions between them. As all of these biological networks commonly are complex networks, they share some properties, as the scale-free topology, the small world effect, and they are not purely regular, not purely random, okay? They would be here. And as they follow a scale-free topology, they have a power law distribution, okay, of nodes. So what this tells us is that there are few nodes that are highly interconnected, and that we know after the name hubs, okay? These hubs would be the very key important genes, let's say, in a genetical network that are related with many other genes, and the other nodes that are not as much related would be less important in the network. Well, here you can see some of the biological networks that we have already said. Protein-protein interaction networks. Protein-protein interactions are physical interactions between two proteins and are quite important when we talk about regulatory, not regulatory, but let's say, what's the word in English? Yes, like the correct functioning of the cell when we are developing any process in the organism, okay? And then metabolic networks with the different kinds of metabolites and the enzymes codifying those paths of processes. And then the regulatory networks that could be, for example, a transcription factor network. A transcription factor is protein that is usually a protein that is regulating the expression of other genes, and then it can be up-regulating or down-regulating, for example, and it will depend how many copies of the gene we have, and that can lead also to pathological stages, okay? Then we have also RNA networks that are also derived from the expression of a gene, of the different genes. If we are regulating a gene, we will have less copies of RNA, and if we are up regulating a gene, we will have more number of copies of a gene. Okay. Here you can see a representation of different biological networks. These networks are all from yeast. They are not humans, but it could be, okay? The first one is from a transcription factor binding network that is obtained from a chip of chromatin immunoprecipitation analysis. This one is from protein-protein interaction networks. This one is for my English, phosphorylation network, and this one is for metabolic network. This one is for a genetic network. Okay, if you are curious on these things or you want to know more, just write me and I will send you some more information. But as I said, we will study a little bit more the biological data and the bioinformatics around this the next session. Okay, let's go into deep of what is interesting for us today, that is the human disease network. I would first like to ask you if you have heard about this concept before or is the first time. I mean, maybe Alejandro mentioned it, but did you know before about it or have worked in any time with these kinds of approaches, or is your first time?
To myself, I think it's the first time.
Okay, great.
For me also.
Okay. Abby? Okay.
Yes, it's fine.
Okay. Let's continue. Neither of you know about this. Let's keep it slow. What is a human disease complex network? The term complex is for complex network to define that the topology of that network is that is a complex network. Okay. Also, human disease is for human diseases. We can find also this type of diseases networks for other type of organisms. But what is interesting for us is the human disease complex network. And since the human genome project that was carried out like the past decade, I think, more or less, that thousands of laboratories and the research community in general was making effort to sequentiate and to understand all the genome of humans. This was developed. I mean, when all of this project was finished, a lot of information was able and a lot of approaches were thought. So the first time that the term human disease complex network was coined was in 2007. It has been some years now. By some researchers in a paper from Proceedings of National Academy of Science of the USA. And HDN is a graph where diseases are related to other diseases based on the similarities that they have with the other diseases, based on the, let's say, relationships established between them. Okay? Such similarities are computed under the assumption that the diseases have shared characteristics. Go et al., that are the authors of these papers from 2010 that I say, based all of their approach in disease genes association, that are those diseases that maybe they have a mutation are causing a disease. Those genes, that when they have a mutation, they cause a disease. This is one of the multiple approaches possible, and it was the first, but as we will be seeing today, there are multiple approaches. This is the first human disease network, the one that was published by Co et al., and all of the subsequent studies in this field were based on this one. The network that you can see here, because the network that we will be interested most is this one. It was originally built under a bipartite graph, that is this one, okay, in which we have two kinds of nodes. In one side we have the diseases, where each node is a different disease, and in the other axis we have genes as you can see, the different genes that are here. Only we establish a connection, only we draw a link between a disease and a gene. If a mutation in the gene was causing the disease. Okay, so I will repeat. We have diseases here, and here we have genes. The gene in one side is linked to the disease in the other if we have a mutation in this gene that is causing this disease. This bipartite graph was called the disesome, and as I said, is the most important, let's say, bipartite graph linking one type of nodes with diseases. What they did later was to make two projections from this bipartite graph. They make the human disease networks where they project the diseases, and they established the links between diseases based on the numbers of genes that were related to that disease of that two diseases shared genes. Okay, let me explain it again. From the disesome, we obtain the human disease network where the nodes are diseases, and the link between diseases is established based on the number of genes that both diseases have associated in the disesome. Okay, I mean the intersection, the number of shared genes. Okay. When we talk about the shared genes, is because that gene is causing a disease when it is mutated. Okay. They also projected another network that is the disease gene network, where the nodes are the genes and not the diseases, and the genes are linked if they have diseases in common. Okay. Also, the width of the link is established based on the number of shared diseases in this case and genes in this case. Here you can see the human disease network complete. Okay, its node corresponds to a different disease and is colored based on the class where they belong. I mean, I think this was made with MES class. Okay, MES. I don't know if you know. We will have a little bit more of a study of what is MES in the fourth lesson that I will give, like in 3 months or so. As you can see, the colors of the classes of diseases pretty much associate with what is the topology of the network. I mean, they are all more or less topologically in the same place based on their class, and also the size of the node is based on the number of connections that it has. So a bigger node means that is linked to many other diseases, and that is what we call a hub. Okay, that in this power law distribution that have a complex network following a scale-free topology are very few hubs, but those nodes that are in the hub are very much interconnected with other nodes. This one is the disease gene network, the gene, because the other one was the HDN. Okay? Well, as I said, the different nodes are the genes, and they are connected if they share diseases, if they are implicated in the same disorder, let's say. Also here, the same as I told you in the last slide, that the colors are based on the the categorizations of diseases. So sorry. Okay, each node that is linked to a class is colored by the class of the disease that is linked to. Those nodes, sorry, that are linked to several classes, disease classes, are colored in gray and set like unclassified. So let's re-summarize what we have seen here. It's just an approach where we have diseases as nodes, and diseases are connected if they share any of the genes that are implicated in causing that disease. Genes are one of the facts or traits that can help us establishing the relationships between diseases, but are not the only ones. Okay, there are multiple features that we can consider when establishing those edges in the network. Later, in 2014, Fu et al. Published a paper in Nature where they first proposed the human symptoms disease network. This approach was a step forward in the field of network medicine and is another way of solving, let's say, some of the complications that the other type of networks weren't able to explain, as if disease is not caused by just a mutation in a gene. Okay. Okay, here you can see the skeleton, the schema of the network that they built. And what we represent in the links here is the number of shared symptoms between the diseases. So here, instead of saying a disease is connected to another disease if they share mutation in one gene, we say a disease is connected to another disease if it has symptoms in common. Also here, as it was in the Human Disease Network, the size of the edge is based on the number of symptoms shared, ¿vale? Okay, sorry. And yes, that's the network that they built, but they didn't build only this network. They also built another one. This one is the one that we were seeing, the one that links diseases based on the symptoms, and that establish the width of the link based on the similarity of their symptoms. But they also built another network that was based on genes and PPIs. Here you can see the different diseases connected by the links of genes, as it was in the Human Disease Network, and then in green you see the PPIs of first order, and in red the PPIs of second order. What we say with first order and second order is if we have inferred that knowledge because two proteins interact together and another two proteins interact together, if one of the proteins in the first interaction is interacting with another of the proteins of the second interaction, we can say that the other two proteins in the two different interactions are also interacting. Okay, it's just like not in silico, but computational approach. What they see with this approach is that when two diseases share symptoms, they also tend to share or genes or PPIs. So it was like a correlation that they could establish that phenotypical or symptom similarity is correlated with a genetical or PPI similarity between diseases. So we have seen the human disease network where they link genes, the human symptom disease networks with they link based on the symptoms, and now we are going to see other disease networks that are based on other multiple traits, not only genes or symptoms when linking to diseases. This was a figure that derived from the first paper, from 2007 of the Human Disease Network, and it says that from the DISSOM, that I remind you is the one that is a bipartite graph where we have the diseases in one edge and the genes in the other, and we establish the connection if only if one mutation in the gene is causing that disease. So well, from that DISSOM, another type of disease networks can be built based on, for example, the drug link between diseases. So if a drug is associated to a disease A and a drug is associated to a disease B, we establish links in the network. The genetic link is the one that we already talked about. Interactonic link is any of the interaction processes that are being carried out in the cell. If two same processes are attached with different diseases, those diseases will be having a relationship. Then the expression link that comes again with the idea that differential expression of genes in different tissues, for example, can be used to build diseases networks and also give us relevant information, because maybe a disease is related to a gene, but another disease is also related to the same gene. But the first disease is caused when expression levels of that gene are upregulated, so we have a high number of copy numbers of that gene, but the other disease is related to the gene when the gene is being downregulated and we have very low numbers of the RNAs related to that gene. So the connection here would be another type of connection if we have down-regulated and up-regulated diseases, let's say, based on the regulation of the genes they are associated to, that if the expression is the same for both diseases, for the gene for both genes, the gene for the gene causing both diseases. Sorry. Also metabolic link when we talk about the metabolites that are in a process. Let's see later, and the phenotypic link that is related with symptoms, the phenotypes that you are showing, that most times we call them signs or symptoms, and also the environmental factors that can be inputted in the organisms. And one that is not here, well, two are microRNAs that we will see later, and comorbidities. comorbidities, I guess you know what they are, but just in case, when you have a disease, a primary disease as cancer, for example, and then you develop another disease, you say that those diseases are comorbidities because are being at the same time in the organism, and maybe they are influencing between each other. The disease one is producing some kind of effects or dysregulating or good regulating in some kind. They are contrasting the bad effects of both diseases. So that's also a very interesting point of to be studied about diseases, and that can be used to build this kind of networks. The first one that we were saying is the metabolism-based human disease network. That if you read in deep the paper from Go et al., the one of the human disease network, you will see that in that network that they build one of the underrepresented groups of diseases where the metabolic diseases, such as, for example, I don't know, diabetes, for example, some diseases that have to do with the metabolism process of the organism. So they were underrepresented in that paper. That can be explained because they didn't have the link of genes, but those diseases can have the link of metabolites between each other. This is what thought the authors of the paper that is shown here, the one that built the metabolism-based human disease network. In this MDN, let's say network, the nodes are again the diseases that are only connected if the associated enzymes are catalyzing adjacent metabolic reactions. Okay, so for example, if a disease is associated to a gene and that gene is encoding one enzyme, and another disease is related to another gene that is encoding another enzyme. If both enzymes encoded by the genes related to the two diseases, if those enzymes are catalyzing metabolic reactions that are adjacent, so that one metabolite is giving another metabolite by the enzyme 1, and the metabolite is giving another metabolite by enzyme 2. If they are just one after the other, they were establishing the link in the previous network. Okay, so this is how they did, counting also the number of times that this happened between two diseases. Maybe two diseases are associated to multiple genes, and the multiple enzymes that are being codified are all catalyzing adjacent metabolic reactions. So that's why the link are bigger or lower depending on the two diseases that they are linking. So, as you see, for example, coronary artery disease is linked in a very strong way to hypertension. So that tells us that the metabolism between those diseases are quite in the same paths, okay, of metabolism. Another disease network, this one is based on expression profiles. We have already talked about this. This one was the first one that was published around the idea of expression profiles, and here they saw that they could establish a hierarchical association between diseases based on the varieties of the expression of the different functional modules of genes. Here you can see some of the agrupations that they did, and some were basic. Let's say they already know, but they could also make other associations that were not evident. For example, the uterine lymphoma is not lymphoma, lylomyoma with the lung cancers. Okay, that it was something that it hadn't been observed before, and that was studied afterwards in the molecular context. From those associations, they built the network that you can see here, and this one was the first network that was based on expression profiles, and they use it to know the drugs that could be used for one disease to another. We will talk later about that. And also they established significant associations that were present in the hierarchy that we saw before, that are, for example, between the malaria and the Crohn's disease over here, and that is thank you to the network representation. And here, another approach is to build the human disease network based on the interactome. That what is the interactome? What we understand for this term is all the physical interaction between all the components, all the molecular components of a cell. Okay. It can be, for example, proteins and metabolites, is to give a more holistic way of thinking. Okay, so in this one you can see different proteins associated to different diseases that are the multiple sclerosis, peroxisomal disorders, and rheumatoid arthritis. And they could find that multiple sclerosis and rheumatoid arthritis were more similar than what they thought in that time. Okay, all of these interactions are PPIs. Okay, protein-protein interactions. Those interactions reveal that the related diseases could have any other kind of relationships. For example, disease pairs that had overlapping modules, as these ones that we can see here, may have some kind of phenotypical similarities or high comorbidity, that they could be present together in an organism. And also the other way around, those diseases that weren't overlapped in modules presented less tendency of having relationships that would be, let's say, detectable clinically talking, okay? This is another work that was carried out in terms of the interactome, where you can see all of these things out modules that we have been talking. That is, first we build the interactome, we obtain the disease genes to build this interactome and the PPIs. Afterwards, we see which diseases share paths within the interactome, and we establish modules based on that. Afterwards, we can see which pathways are arranged of this when the number of components is so large that you cannot experimentally track all of those genes or proteins that are related to a disease. And afterwards, you have to validate that approach, maybe by annotating the genes functionally with the gene ontology or some tissue specificity. Let's say they are manifested always in the liver or always in the lung, etc. And also the similarity between the phenotypic manifestations. Okay. Also, you can see the homogeneity in the co-expression between the different modules and the interactions and the response to drugs. And finally, what you want from this kind of approach is to see if you can obtain new candidates for disease genes, that is, those genes that are causing diseases. Also to say, to identify which pathways are the responsible of diseases. Or, for example, know which targets of drugs you can use for a disease, which specifically complex you have to work to fight against, let's say, to develop a drug. Okay. Here you can say that all of the human genes that were known in that time, that was 2011, only about 1,700 were associated to specific diseases. From those, some of them were known to be essential. I mean, to be essential, like if you don't have them, it is lethal for the embryo. You cannot develop some of the key functions that you need to survive. Okay. So here you can find that only these ones are essential disease-causing genes. In the second figure, we can see that the difference between those essential disease genes and those that are not essential disease genes, so I mean between this one and these ones, are that normally essential disease genes tend to group, to cluster together in the lysosome, but the non-essential disease genes are segregated into not have spaces of the network. Okay. There are many ways to let's say classificate the nodes in the network and establish modules. We can find topological modules that those are based on the network properties, the modularity, the centrality, all of the metrics that you studied. But those are mathematical modules. Okay. Then we can find functional modules based on annotations, for example, as we said, the geo annotation, the gene ontology annotations, based on the biological processes in which the gene or the protein is being involved. And then we have the disease module that is what it is interesting for us in the DSSOM, that represents a group of nodes whose perturbation, that is mutations, deletions, copy number variations, can be linked to a particular disease or phenotypes. Okay. Sometimes they are the same. I mean, topological, functional, and disease modules can overlap, so that functional modules correspond to topological modules, and disease can be shown as the breakdown of a functional module. Okay. Another one that we have already mentioned is the human disease network based on comorbidities. This kind of networks are built based on the patient medical history. So if we have a patient that comes to admission to a hospital and it has two diagnoses, what it means that it has comorbidity because both diseases are present in the person at the same time. We then discharge this patient from the hospital. It goes back home, and then it comes another time, and it has other diagnoses at the admittance. We can model this kind of networks. Where, for example, if we had a patient that it is diagnosed the first time with disease 1 and disease 2. Later he comes and it is diagnosed with disease 2 and disease 3, and later with disease 1, later with disease 2 and disease 4. This network is built for the transitions between the different diseases, okay? And for example, we count the different nodes based on the difference between the diagnosis that we give the patient in the distinct admissions to the hospital. So in this case, we have to transit first to the disease 1 stage. So we have for disease 1 one transition, then it transits to disease 2, not 2, but we don't have it, so it is another transition. We have two transitions, and then to disease 1 again, okay, and then to disease 2, we don't have it again. So from disease 1 to disease 2, we have 3 because we first have here disease 2, so we have 1, 2, and 3, okay, so we have 3. And then to disease 2 to disease 1, we have from here to here. I don't know if I am explaining myself. If not, just stop me. And then we gave the node size also a metric that is the prevalence, that is the times that the person has this diagnosis. So this network is built for its patient, as you can see here, and it is summarized in one network. I didn't mention that these kind of networks are built for particular diseases. For example, in this case was for type 2 diabetes, and they track these kind of networks for both disease, a cohort of disease of patients having this disease and a cohort of patients not having this disease but other diseases. And they make two networks and they summarize it in this one, and they finally were able to build this kind of network where we can see the relationships between the different diseases based on the transition stages between each other and in the two different populations. This one is for the population that was having diabetes 2, and it is different from the one that was not having diabetes. They could learn some things that weren't able apparently to the research in diabetes. And also you have to understand this as uh, let's say 3 states of researching in a specific disease, how we can look for other similar molecular cerners, let's say. These kind of networks can also be built based on social network analysis, for example, in Twitter, but they are not as reliable as if we take the medical history patient records. Here you can find another human disease network. This one is based on drugs. The gold or yellow nodes are representing drugs, and the blue-green nodes are representing diseases. And they built this network also based on the expression of the genes, and it was the one that was the first to give this idea of targeting different genes, proteins, let's say, when you know the association with a drug of a disease. Okay. Another one that is for human disease network based on drugs, and they model different processes with different networks. They predicted some associations of drug sharing between diseases. So I'm not sure if they later could use all of those drugs for that diseases, but it was a good starting point to research on those drugs. Okay. This one is for microRNAs. MicroRNAs are a special type of RNAs that regulate some functions of the cell, or some types of signaling and those stuff that have been seen to be very specific of some diseases. So this one is quite recent. It's from 2019, and as you can see, they established some connections that were related because of microRNA mediated processes. We will see another slide with with this approach later because they built also a huge like approach, not approach, but study of how diseases networks can be built based on these microRNAs, and I think is a good important application case. So we will see it later if we have time, because I think I am being very slow explaining things. I wanted to just to make clear that when we study disease networks, there are lots of layers that we can take into account. Here, for example, you can see in the down layer metabolic networks, where we can find regulatory networks, protein-protein interactions in yellow, and metabolic itself networks, where the different metabolites are transforming one to another. And then we have another layer that can be the social network, how we link between other person, and the how is our family, or the physical proximity with another people, or whatever, that can also be used to, for example, nowadays in the epidemiological context, can also be used to learn patterns of diseases. It's like an abstraction that can all be used in the development of disease networks. Although these networks are independent, we can project its information, and we can always have more insights when studying diseases. Okay, yes, we have 20 minutes, and I think I wouldn't spend them all. Some approaches and utilities of these disease networks. To summarize, we can obtain a larger and better understanding of the causes and effects of diseases. We can get more insights of the patterns that are not apparently to the individual study of each disease. We can also take the approach of drug repositioning that we will comment now, and identify new targets for treatments or biomarkers to be able to say if one person has the disease or not. Also, as I already said, these new classification methods of diseases that are more suitable and that more accurate than the ones that we already have. Okay, drug repositioning. I think this is one of the most interesting topic related to disease networks, and the one that has no future. And I don't know, one of the things to do this, okay, I don't get the word, okay, doesn't matter. This all started because they established this hypothesis that can be found in disease similarities. That is, if you have a drug X that is used to treat disease A, and we found that disease A and disease B are similar, let's say in context of similarities from the symptoms or the biological components, it would be possible that drug that drug X can be used to treat disease B. Related to this, we find that the number of drugs has been developed, has been decreasing through the years. As we already commented, all of the clinical trials and all of the studies that are performed by the pharmaceutical industry are more and more restrictive to have safety procedures and safety approved drugs. I mean, not everything is going to be approved, only those that are that we are sure that are highly or completely safe. This is related to drug repositioning in the way that the knowledge that we have now can be restructured to get treatments by the already drugs that we have. So, drug repositioning is to use already existing drugs for new diseases so that we can evade the time, risk, and cost that are derived from de novo drug development. That is why we have a less amount of approved drugs in the recent years. Those time, risk, and cost that are associated to de novo drug development. As I said, drug repositioning is the application of known drugs and compounds to treat new indications. And the advantages of drug repositioning include less cost and less time to approve a new drug and put it in the market. This is the graph representing on one side the evolution that it takes the process of drug development in de novo approach, and this one is the one of drug repositioning. Okay, as you see, you can skip this part of the process, and that way you can evade the risk and the cost, also the time, the most important time, that it is from 8 years, from 2 to 3 years, okay, more or less. This is the representation of the number of articles in Google Scholar that contain the term drug repositioning during the last year, well, till 2017. But as you can see, is a tendency rising a lot, and that's because it has become really important and popular, not popular, but relevant for the scientific community to analyze this type of approach, that is drug repositioning. So this for drug repositioning now. This is similarity computation. This is one of the derived problems or challenge that you have when you build disease networks. Okay. All of this process is underpinned by what is called vector space models and methods that treat diseases as vectors of features. For example, if genes are the ones that are being studied related to diseases, we will build a vector of the different genes that are associated to diseases. I represent here a bidimensional space, but it's just for you to clarify, but this won't ever happen because we will have thousands of genes, what would lead to thousands of dimensions. Then, when we have represented our disease as a vector of features, we can measure the similarity between diseases based on different metrics that are already vector operations, as can be the cosine similarity, that would be this angle of here. Also, we can calculate Jacquard similarity, but that would be the intersection between the union of elements in both vectors, and also the dissimilarity, that is quite similar to Jacquard, but not the same exactly. For example, this is just one silly example calculating Jacquard similarity for two diseases. That would be disease 1 and disease 2. Disease 1 is associated to the vector A of genes because it's related to gene 1, gene 2, gene 3, gene 5. And disease 2 that is associated to vector B of genes because it is related to gene 2, gene 4, and gene 5. Then we will have to compute the intersection between the two vectors. That will be the gene 2 and the gene 5. So we have two set genes. And afterwards, we have to build the union of the two vectors. That would be the 5 genes that are here. So the Jacquard similarity of the two diseases, disease 1 and disease 2, would be 2 between 5. Okay. There are multiple approaches when calculating the similarity between two diseases. I have just pointed out three very easy metrics, but we can build similarities also based on, for example, the functional annotations that we have been talking about, or the different sub-graphs that are derived from the complete graph, and other many that you can see here that I'm not going to extend much, but I just wanted to point out that there have been a lot of research in this topic. Okay. Also, what we can do is not treating diseases networks as different approaches when talking about one feature or another. I mean, for example, the thing that we see that we can relate diseases based on drugs, but also based on the expression profiles, and then join it and see which relationships are established based on the both. Okay, this can be seen as a multi-layer network. A multi-layer network is just a network that has different levels of diseases. Relationships. I mean, it can have the same diseases in the different layers, but different relationships based on one layer or another. This particular network is based on the phenotypical relationships between diseases and the genotype relationships between diseases. You can see that here is represented as the different phenotypes, the different diseases in pink, and the different genotypes in blue. Some of the relationships are both in the phenotypical and the genotypical layer. In that case, we call it overlapping link. But the interesting thing here is also that there are communities that are present in one layer and another. What I mean is that are communities that are the combination between the different relationships established in one layer and in another, and that cannot be interpreted only with one layer, but it has to take the two. Okay, and that is what is very important in this kind of approaches. I already mentioned this, but for example, disease networks can help us establishing new disease classification systems, more accurate than the ones that we have, that you can see here. For example, ICD, MAIS, SNOMED, UMLS, TO. We will talk about them later. Later, I mean, in 4 sessions. But all of them have been built based on different criteria. This one is because of statistical complaints or something, and this one is for the papers in PubMed. And the other ones for different reasons that we will be seeing deeply, but not now. But all of them have been seen to lack of molecular information, for example, and disease networks would be one of the approaches that can give us more insights in this. This was already established by the National Academy of Science in 2011, and efforts are being made towards these new classifications. This example is one of the clustering diseases model that we have already done for our project, and what I wanted to know to tell is nosology groups, what it means. Nosology groups is just the different etiology classification systems that we have for diseases, okay? And I think I'm going to stop here, and I will start next day just with the application case of DISNet, because I think it's relevant to give a little bit more of time, and I will just mention the other application cases that we have. But I think I was very slow explaining. I think I have repeated myself too much. I don't know if you have any question or something that you would like me to clarify, just tell me, please.
No, I think it's fine for me.
Yeah, for me too. I need to read it again before the next class, but it's okay.
Sorry?
I need to read it, that presentation one time before the next class, but for the moment I think it's okay.
Okay. Okay, great. So basically like that. Next day we will start with DISNet, and we will then devote a little bit more in biological data and these bioinformatic tools and approaches. If you have something else that you want to ask, you can also write me like email or anything, okay, or by Teams or whatever is fine to you. Yeah.
Okay, thanks.
No problem.
Just a question. Do you know, next week we are not available, the three of us, because we have summer camp. So we are not available to be in class.
You won't be able to be in the class.
Yes.
I'm going to ask Alejandro, but one thing that we can do, if that's fine for you, I can record the session and upload it, and you can check whenever you want.
But Abinaya and Carlos too, because they are in the bookend too. So the three of us are not here.
I think we can do it. Okay. Yeah, no problem. I will talk to Alejandro though, but
Yes.
I think that's fine. So I hope I have translated the important keys of human disease networks, because I really like this stuff. We have not going into practical things, but I mean, they are networks. You can work with them as networks with Python, with R, also with visualizing tools as it can be Citruscape or Gephi or any other. Okay, So yeah.
Great.
So see you not next session. Carlos, yes, but not the other. Carlos, you will be able to come next Tuesday or not?
No, I'm also in the boot
Ah,
camp
okay,
next
the
week.
three of you. Okay.
Yes.
No problem.
Yeah.
I will record the session. No problem. Okay. So have nice 2 weeks.
Thank you. Have a nice day.
Okay. Have a nice week.
Thank you very
Thank
much.
you.
Bye bye.
Bye.